All right. Well, hello, everybody. It's my great pleasure to be able to introduce Dr. James Bradley today as our C-DEBI speaker. James came to USC in 2016 from the UK as a C-DEBI postdoctoral fellow to work with Doug LaRoe on modeling in marine sediments. And during his 2 years or so here in Southern California, he published 4 excellent papers, 3 of those as first authors, so a highly productive postdoctoral fellowship, and just had a 5th paper rejected at Science, which I'm sure he'll talk a little bit about today, but we're reworking that one to get resubmitted in the very, very near future. He's now on the faculty at Queen Mary University of London, and today he'll be talking about the power of microbial life in marine sediments. James, take it away.
Thanks very much. I'd just like to start by thanking the C-DEBI community a huge amount for such fantastic opportunities throughout the 2-year postdoc and continuing in the present day, and of course in the future as well. Before I get started with my talk, I'd like to also thank and acknowledge my co-authors of this study. So that would be Doug LaRoe, Jan Amend, Sandra Arendt, Eva Bovics, Andy Dale, and Matthias Egger, and also acknowledge the funding agencies for this study, particularly C-DEBI. So I'm going to be talking about the power of microbial life in marine sediments. When we think about the nature of life on Earth, we're most familiar with life at the surface of the Earth, and that's because we have a whole range of techniques to make direct observations and measurements of life surrounding us. We can even use satellites to get a kind of global picture of the various biomes that make up the Earth. But if we think about subsurface environments, our understanding is hugely limited to opportunistic access via boreholes and mine shafts and access to the subsurface, things like that. And what I hope to do in this talk is to fill in this black box a little bit. So we do know that the subsurface contains life. Scientists have gone aboard drill ships such as this one and drilled down underneath the sediment water interface at the bottom of the ocean and collected cores from marine sediments. And analyzing those cores, they found a huge abundance of microbial life. So cell numbers ranging from 10 to the 4 to 10 to the 8, 10 to the 9 cells per centimeter cubed of sediment as you go down beneath the seafloor. More recent upscaling of those figures has resulted in an estimate of roughly 3 times 10 to the 29 cells contained in marine sediments globally. So that's roughly equivalent to the total number of cells in the world's soils and also in the world's oceans. But compared to soils and oceans, we know comparatively little about the marine sediment environment. We do know, however, that these organisms are active. We know that by various means, but you can see here in this photograph, the scientists carrying this core of sediment are wearing gas masks, and those gas masks are to protect themselves from the hydrogen sulfide gas that's given off as a byproduct of these microbial metabolisms. We also know that these organisms are persistent because they can survive over geological timescales. These organisms are also hugely important in our understanding and in driving the degradation or the burial of organic carbon in marine sediments. So they exert a huge control on the global carbon cycle and have a bearing on the transfer of carbon from the atmosphere into the oceans and into marine sediments where it's stored over very long timescales. So taking our global data sets of the number of cells in marine sediments, you can start to get at a picture. One of the things that is shown in this graph is the decreasing concentration of cells as you go down further, deeper into marine sediment. So decreasing from 10 to the 5 to 10 to the 9 cells, going down meters or hundreds of meters, even kilometers below the seafloor. Another trend in this graph is the distinction that it really matters where you drill. So depending on where you drill, you have different concentrations of cells. But as you go down into the subsurface environment, drilling even deeper, you're essentially traveling back in time. So we can equate these different depths to age. And here we have a very clear trend of decreasing cell concentration with increasing age of marine sediments. So in sediment, which is hundreds of years old, we have tens of millions of cells per centimeter cubed, all the way down to only hundreds of cells surviving in sediment that is tens of millions of years old. Even these organisms that are in sediment that is extremely ancient require energy in order to stay alive. So they get that energy from catalyzing exergonic, that is energy yielding redox reactions at some rate to provide power. We're going to be talking a little bit about power in this talk. Power is basically energy per time. So you can think of power in the unit of watts, that is joules per second. And organisms use that power in order to fuel endogonic, energy requiring reactions in order to carry out maintenance, in order to synthesize biomolecules, and in order to, in some cases, to grow. The primary electron donor in marine sediments is organic carbon. And when you plot organic carbon on this graph, you see another trend, which is a very clear decline in organic carbon from sediments which are relatively young, containing lots of cells, all the way down to very organic carbon deplete sediments that is millions of years old, containing very few cells. It's really important to be able to think about microbial energetics in this context, because really it governs a huge number of factors that are important to this biosphere. So the interaction between organisms and energy will tell us whether or not these organisms are able to grow, whether or not they're able to adaptively evolve or assemble in different environments. Understanding their energy utilization gives us some insights into the lower energy or power requirements to life. And the interaction between these organisms and energy also governs organic carbon degradation and the global carbon cycle. What you can notice on this graph is with increasing time since burial, there is net decrease in biomass. So this is a system where there is net death. So there is a huge production of dead organisms. And interestingly, dead organisms have been shown to be able to sustain living cells in a lab environment for many months, even many years. So organisms studied in this nature have exerted a kind of cannibalistic response, using DNA as a nutrient and regulating their death and their life in order to be able to sustain a long-term, slow-growing population. So a major question was, can necromass, the dead remains of living cells, support living cells in energy-starved marine sediments where there is a huge generation of dead biomass? Unfortunately, we can't use lab incubations to get at the answer to this question because they're simply too high energy. When you take an organism in a lab environment, it's recently been growing in a culture, but the organisms in marine sediments have been sat in that environment in this stationary state for tens of millions of years. So in order to get at the answer of this question, I developed a kind of new bioenergetic modeling approach, comparing the power demand of these organisms, which is calculated by estimating per cell power demand and a function that will tell us how much biomass is in an environment, and comparing that with the power supply. And the power supply can be calculated by knowledge of the Gibbs energy of a reaction and the rate at which that reaction is taking place. So we did this for the South Pacific Gyre, which is indicated where these white circles are. It's a highly oligotrophic part of the ocean where we think that the sediments underlying that ocean provide very little energy to the organisms within it. So we use a various suite of models to be able to derive functions that will predict the change in microbial abundance with increasing sediment age, so increasing burial. And then we can use the trends in that model to predict the rates of necromass production. So in the number of dead cells that are produced per year per centimeter cubed of sediment. And that will also give us a death rate per year. And when you compare these mortality rates that we're deriving from this modeling approach, 10 to the minus 7, 10 to the minus 6 per year, these are at least 3, 4, 5 orders of magnitude lower than mortality rates that we're used to thinking about for surface environments. So for when we're modeling soils, microbes in soils on the surface of Earth. We can use energetic calculations to tell us how much energy is given off when we, or is available to organisms when we oxidize the various components of an organism using oxygen. So our South Pacific gyre sediments have oxygen available. So we use oxygen as an electron acceptor when we are degrading these biomolecules as a source of energy. And you can see that the nature of the biomolecule really does have an important bearing on how much energy is available. So for example, though amino acids make up over 70% of a cell, they only produce about 60% of the energy, whereas fatty acids, comprising a very small proportion of the biomolecules that comprise a cell, produce a huge amount of energy when it is oxidized in the presence of oxygen. So for our South Pacific gyre sediments, we can use this modeling approach to tell us how much power is available from the oxidation of microbial necromass. So the scale here ranging in power of nanojoules per year over several orders of magnitude. The variability in this is due to the variability in cell size. And then we can use the other half of the model to tell us some estimate of what the demand for energy of the microbial community that is living in these sediments is. And there's a huge amount of uncertainty here, but a clear thing to take away from this graph is that necromass does not provide enough power, despite being produced widely in marine sediments to support the demand for the living microbial community. If we use the same approach to model the amount of energy or power produced by the oxidation of organic carbon, we can in fact see that it does provide, we think, enough power to just about support the demands of the microbial community. So it's likely that these organisms are interacting with organic carbon that is buried within these marine sediments and using it as a source of power in order to stay alive for millions of years. So this study comes into context when we think about energy in subsea floor habitats. What we do know is that the amount of energy available to organisms does vary from one location to another. And because of that, it imposes a significant limit on the activity and survival of microbes in marine sediments. A recent study by Yuki Morono and his colleagues have shown that organisms from incredibly ancient settings can in fact be revived when they're fed isotopically labeled substrates. These organisms, this is a groundbreaking study because it really shows that these organisms that are found in marine sediments are living and are capable of being reactivated. So when we think about trying to model these organisms, we need to use a new kind of approach which is able to account for microbial activity that is not just growth. So as part of this C-DEBI postdoc fellowship, I developed a new model which actually describes how microbes interact with their environment in marine sediments in a way that doesn't require them to be growing to be using energy. So actually accounting for the different stages of activity or states of activity, all the way from active through to a dormant state, and interacting with energy in a way that can provide enough power to synthesize molecules just for maintenance rather than for growth. If we think about how much energy or power it takes to keep organisms alive, a human runs on about 100 watts of power. So that's roughly 2 light bulbs. And it's been estimated that an organism from the deep subsurface environment runs on about 1 to 30,000 zeptawatts of power. And a zeptawatt is 10 to the minus 21 watts. So the organisms in the subsurface are interacting with energy on a fundamentally different way to life as we know it on the surface of the Earth. But outside of a few very site-specific measurements and studies indicated by these dots, the level of activity of subsea floor microbes on a global scale is virtually unknown. So what I've set out to do recently is to try to quantify the power of subsea floor life on a global scale to get at the answers to some of these questions indicated on the right. Can organisms in marine sediments, do they have enough energy to be able to grow? If they're growing, do they have enough energy to be able to kind of adaptively evolve or assemble according to their environment? Can we use this kind of study to probe what the lower limit to life is? And by quantifying the power or the activity of organisms on a global scale, can it tell us something about organic carbon degradation and the nature by which organic carbon is attacked by organisms in the subsurface environment? So in order to get at that question, I developed a new global ecosystem bioenergetic model where I could calculate the power per cell based on this relatively simple reaction, this very relatively simple equation, where we have the power per cell equals the rate at which a reaction is taking place multiplied by the amount of energy that is yielded from that reaction divided by the number of organisms that are carrying out that reaction. So I'm going to walk you through very briefly the various components of this equation, the first component being the rate of reaction. So we use a global POC degradation model implemented on a 1/4 of a degree by 1/4 of a degree grid to predict the change in organic carbon, that is the degradation of organic carbon on a global scale as a function of sediment age, but using a conversion between sediment age and sediment depth. We implement this model using a reactive continuum approach, using a varying number of parameters to describe the reactivity of organic carbon and model degradation of organic carbon within a bioturbated zone, within Holocene age sediments, and all the way down to Pleistocene age sediments, so roughly 2.6 million years ago. To get at the free energy term, we need to know what that organic carbon is being oxidized with. So we need to be able to predict, first of all, what the global oxygen penetration depth is, and we will assign any organic carbon that is degraded below the sediment water interface but above the oxygen penetration depth to aerobic heterotrophy. So in order to predict that depth, we use a relationship between the oxygen penetration depth and water depth developed by Ronnie Glood and others, and combine that with a global model to predict the distribution of oxygen within marine sediments on a global scale. Beneath the oxygen penetration depth comes the sulfate-methane transition. So we use Matthias Egger's model describing the variability of the sulfate-methane transition zone according to sedimentation rate and some other factors in order to predict the global distribution of the SMT. And wherever there's white in this image indicates that there is no SMT present in this setting. So that means that either oxygen or the sulfate zone, the sulfate producing zone, penetrates all the way through to the basement of the sediment at this location. And then below the sulfate methane transition zone, we prescribe all of the organic carbon that is degraded to methanogenesis. So then we can use energetic calculations to estimate what the Gibbs free energy is per mole of organic carbon degraded within each of these zones. And finally, for the denominator of this equation, we need to be able to predict what the cell abundance is for every location on Earth. And thankfully, Jens Kollmeier and others have developed a global model that predicts the distribution of microbial cells in the subsea floor environment. So essentially what we do is we take this model and we put it onto our global grid, our 0.25 by 0.25 degree global grid, in order to get at this information. So by using this relatively simple looking approach, where we have the rate of reaction multiplied by the Gibbs energy divided by the cell abundance, we can simultaneously quantify a huge number of things on a global scale. First of all, the global distribution of electron acceptors. So we are predicting by this approach that oxic sediments comprise a very small proportion of the volume of marine sediments compared to sulfate reducing and primarily methanogenic sediments. We can also predict the distribution of these electron acceptors for any depth or age slice. So for the sake of this talk, I'm showing you a slice of equal sediment age at 21,000 years. And you can see that oxic sediments are generally located on the drier settings, so further away from the continents. And an interesting thing to note is that although methanogenic sediments comprise a huge proportion of the volume, in terms of where they're distributed, tends to be only very close to coastlines. But the reason why the volume is so big is because these sediments are incredibly thick near coastal regions. We can also use this model to quantify the distribution and the rate and the thermodynamic properties of organic carbon that is stored within marine sediment. And you can see that this generally reflects the volume and also of the organic carbon that is degraded within marine sediment. So I'm showing you here in gigatons of carbon per year. And the distribution of by which electron acceptor the organic carbon is degraded is starting to vary slightly from where we see the distribution of electron acceptors and POC stored. We can again plot this for any depth or age slice, and you can see that primarily organic carbon is being rapidly degraded, again, near productive coastal regions. We can use Jens's model to predict the global distribution of cells, and when combined with the distribution of electron acceptors, we know roughly what kind of setting these cells are sitting in. And again, this reflects roughly the volume and the organic carbon that is stored in marine sediments. We would expect this because typically where you have high organic carbon concentrations, you also have high biomass concentrations. So it's encouraging that our cell abundance reflects the POC that is stored in marine sediments somewhat. And again, we can map this distribution. And then finally, talking about power, so the reason why we're doing this study in the first place, we're predicting the global utilization of energy, essentially power. And you can see by the pie chart that actually the distribution is very different from the amount of organic carbon that is stored or degraded, or the distribution of the cells or electron acceptors within marine sediments. So we'll go a little bit in more detail about this distribution of where organisms are using energy. But if we sum up the total amount of power that is produced or utilized by microorganisms in marine sediments globally, we get around 40 gigawatts of power. And that is roughly equivalent to the power output of Denmark. But to tie it back to subsurface life, it's about 0.1, it's less than 0.1% of the total power that is produced in the marine photic zone. So this just goes to emphasize that the subsurface biosphere is, although it receives most of its energy is derived from surface environments, from the production of organic carbon in the photic zone, and then that sinking through the water column and being buried in marine sediments. By the time it gets there, this biosphere is hugely depleted in terms of the energy that is processed by the cells. So if we think about mapping that distribution of power onto our oxic sediments and our sulfate reducing sediments and our methanogenic sediments, you can see oxic sediments generally are at the higher end of the power spectrum, so between 10 to the minus 18 and 10 to the minus 16 watts of power per cell. Our sulfate reducing sediments are generally, you can see by the cooler colors, are generally more power limited. So 10 to the minus 20 to 10 to the minus 18 watts per cell. And methanogenic sediments are at our lowest part of the power spectrum. Again, the white areas of these graphs indicate where you do not have methanogenic or sulfate reducing sediments. So our methanogenic sediments are the lowest in power, but they're also generally restricted to coastal regions. And that is actually a really interesting finding, is that we have some of the higher powers in oxic settings, which we traditionally thought were very energy limited for marine sediments, but on a per cell basis provide somewhat higher powers than coastal settings, which seem extremely energy limited for organisms. And actually, when it comes down to it, you do have more carbon that is stored and degraded in coastal settings, but you have many, many more cells. So you have a huge number of more mouths to feed with that organic carbon. In our oxic sediments, where we have relatively higher power per cell, this is where we do see stable consortia of organisms surviving for thousands or millions of years. Whereas in our coastal environments, although we do have initially a huge amount of biomass that's deposited, this is where we see the most rapid mortality rates of microorganisms in marine sediments. The vast majority of organisms deposited in this environment die, and it is likely because they are starved of energy. So if we map these spectrum of power per cell across this gradient and put that somewhat into context, we're talking 6 orders of magnitude from 10 to the minus 21 watts per cell to 10 to the minus 16 watts per cell. As I said before, a human runs on roughly 100 watts of power. So that goes way beyond the scale of this graph. The lowest maintenance power, that is the power that cannot keep an organism in a growth state, but is only just enough power for an organism to maintain itself, that has ever been measured in a lab culture, is roughly 10 to the minus 15 watts. So that falls where this red arrow is indicating. The lowest maintenance power that has ever been measured in a natural setting falls between 10 to the minus 17 and 10 to the minus 16 watts. So our marine sediment habitat, all the organisms in sediments are below this power. The lowest maintenance power that has ever been modeled for marine sediments falls between 10 to the minus 19 and 10 to the minus 18 watts per cell. And we can see here again that pretty much most of the organisms in marine sediments fall much lower on this power spectrum. So the take-home messages from this part of the study is, hopefully I have shed some light on the global nature of the subsea floor system and how these organisms are interacting with energy. So opening up that black box a little bit. I would argue that the overwhelming majority of subsea floor life subsists under extreme energy limitation. The fluxes of energy that these organisms are experiencing fall far short of what we previously thought were the maintenance requirements for life. And this suggests that growth and thus evolution of these organisms cannot be sustained in this deep biosphere setting. And also this is showing us that sediments really are critical to understanding what the minimum power for life is, giving us some insight into environments where we might look that could support life at powers of roughly 10 to the minus 20, 10 to the minus 19 watts. So I'd like to finish there, and I'll be happy to take some questions. Let me see if I can find the chat box to see if anything is coming in.
Yeah, thank you, James. So yeah, if anyone has any questions, the chat window can be opened by clicking on the sort of cartoon word balloon icon at the bottom of your screens. I guess someone entered a question in the Q&A module. So James, if you want to click that, Jesse had a question for you there.
Okay, so we've got a question from Jesse. Does the composition of the POC, the identity of the organic carbon molecules at different depths, influence the power per cell calculations, or is it only the identity of the electron acceptor? That's a really good question. We do try to account for the different compositions of the organic carbon when determining its reactivity within the POC degradation model. In terms of the identity of the molecule affecting the Gibbs energy term, so the POC model informing the rate of reaction, but in terms of the molecule informing the energetics of the reaction, it certainly does have some bearing on the calculation of how much energy is yielded when you degrade a molecule of organic carbon, whether that molecule be an amino acid or a fatty acid, for example, as we saw in the power calculations before. But that variability can be 5, 10, 15%. And in fact, we're seeing powers that span 5 or 6 orders of magnitude. So although the identity of the molecule is important on a range of a few percentages, it's not going to affect the overall result that we're seeing here, which is powers that vary by, 3, 4, 5 orders of magnitude. So I'm seeing a question from Jans asking if I can say a few words about the other reactions, for example, other electron donor reactions, metals as electron acceptors. So we simplify the reactions occurring in the subsea floor environment into organic carbon that is degraded with oxygen, with sulfate, and to methanogenesis. This is a simplification, and we do know that there are other reactions that produce that organisms can gain energy from in these environments. But some previous modeling work has shown us that actually by simplifying the degradation of organic carbon to just these 3 systems, we get a pretty good understanding of what we capture pretty well, predominantly the vast majority of organic carbon degradation just with these 3 reactions. So if you think about, for example, iron metabolisms, they are extremely important and interesting on an evolutionary perspective, and in driving some biogeochemical processes. But in terms of on a global scale, they don't really factor in to the nature of energetics when we talk about organic carbon degradation on a global scale. If we think about secondary redox reactions as a source of energy, by nature, these are secondary reactions. So in order to occur, they need the primary reaction, which is the degradation of organic carbon, to happen in order to produce the reactants that will be used in these secondary redox reactions. So by their very nature, they are at a large step back from organic carbon utilization. I've got a question from Ben Tully. What do you think the cells are doing that fall below the lowest modeled environmental maintenance energy biologically. So these cells are sitting there and they're dying slowly. The way in which we see the cell abundance changing as we go deeper into sediments, we very rarely see an increase in cell abundance. It's almost always a net decrease in cell abundance, which is suggesting that these organisms are dying. Because we see continued degradation of organic carbon, that means that these organisms are interacting with their environment somewhat. So they could be using the very small amounts of energy that they acquire from that reaction to repair biomolecules and perform kind of essential maintenance at a very, very slow rate to keep them alive. If I wanted to confirm the accuracy of my model, how many global sediment samples would I need and how deep on average? So that's a great question. The reactive continuum model approach that we're using, this approach has been used widely on marine sediments to predict organic carbon degradation rates on the timescale of thousands of years to millions of years. So the approach has been verified previously by doing that. In terms of comparing our model results to site-specific data, in some places the model does very well, and in some places the model might be off by, I don't know, some percentage points, tens of percent maybe. It's worth saying that this is a mechanistic model. So the things that go towards informing the processes that occur within the model are real mechanistic knowledge of the system rather than empirical correlations between data. So because of that, we can be confident that the modeling is doing a reasonably good job and that it's not limited by data. One thing that you miss by just comparing global modeling results to a specific marine sediment is to data from a specific marine sediment core, is that we don't know that that core is generally representative of the much wider area. So it can be encouraging to have a model result that matches up to the sediment core data very well, but because the model predicts on a regional to global scale, there is limited value unless we have very extensive core data that can constrain simulations across a much wider extent than just the width of a core from a drill ship. I've got another question from Jesse. In a premise of your model that all cells and sediments is it a premise in your model that all cells and sediments are utilizing energy? Any sense of a constraint of what fraction of cells and sediments might be legitimately dead, not utilizing any energy? Or do you think that they're all not quite dead, or better, would they then fall apart? Yeah, I mean, the way that I am, the assumption that I'm making is that every cell that is predicted to be there is alive and interacting with its environment in some sense in order to very slowly gain energy. As I described earlier, it has been shown that these zombie organisms can be recovered and take up labeled substrates. So they can be revived and start to grow again. I would have thought, I actually don't know what the lifetime of a dead cell in a marine sediment environment is, but I would have thought that they'd start to be degraded, especially when you're considering the timescale that we're talking about here, which is millions of years. So I do make the assumption that every cell that we find is living, or every cell that's predicted by the model is living. To what extent that assumption is true, I don't know the answer. But if, for example, we say that, okay, only 80% or let's say 50% of those cells are living, that's going to change our power calculations by 50 or 80%, which is not going to cause considerable variation in the powers that we derive, because the power that we derive spans 6 orders of magnitude. So again, the variability in our understanding of cell life and death and how many there are doesn't have a huge bearing on the power calculations, because it varies by such a large amount. A question, what is the estimated lifetime of a dead cell to be detectable using molecular biology, but before it completely disintegrates? Let's not forget about the stability of free DNA for a moment. So I don't know what the lifetime of a dead cell is in marine sediment. It sounds strange to say. It's worth noting that the model is informed by counts of intact cells. So here we're not talking about environmental DNA, which is known to be occurring in marine sediment environments. Here we're talking about sediment samples that have been collected and analyzed, and what looks like intact cells have been found, and this is what is counted. A further question, do you think that there are non-vegetative forms of microorganisms in subsea floor sediments, so spore forms? Yeah, absolutely. And there has been a number of papers that have been showing that spores are known to be widely distributed in marine sediments on local and global scales. It would be interesting to expand this approach to compare the energy requirement of zombie cells versus those of spores that have to, in some sense, be able to sense their environment so that they know when to stop being a spore and start being a living cell again. And we've got a suggestion to look at papers from Casey Kubert at the spore formers in marine sediments in the chat box. That's a great suggestion.
All right, then. If there are no more questions, I just want to thank you, James, for the great talk, and of course to all of you here for joining the seminar. This was our last seminar of the 2019 year speaker series, but we will be returning with 4 more talks in 2020. We're planning on 2 in late spring and 2 in early fall as this year. And we'll be soliciting nominations for speakers soon, so stay tuned to our social media and newsletter for that announcement. Thank you again, James, on behalf of the C-DEBI Speaker Series, and we will be posting this recording shortly to YouTube for those who couldn't be here for the live presentation.
Thank you. All right, I'm signing out.